Introducing Morph, our multi-omics research factory. Morph is a web-based platform for analysing complex biological datasets. It was developed to understand how microbes respond to the stressful conditions of bioproduction with the aim of engineering more robust strains and achieving higher yields. It's been successfully used for industrial biotechnology research and is now being offered as a tool for both academia and industry to store, share and interrogate fermentation and multi-omics data. Morph illustrates the expression levels of genes, proteins, metabolites and lipids and identifies key changes between time points or experimental conditions. Morph is designed to be user-friendly so that scientists can interrogate their data without needing specialist programming or command line coding skills. And being web-based, it can be accessed from anywhere. It's built by biologists for biologists, making bioinformatic data accessible to all. Morph can perform complex data analysis with ease, and it can be used to share published data freely, allowing the community to interrogate the data further, or data can be shared securely within a team. Morph has been used by a variety of national and international project teams and research communities. The BBSRC-funded Detox project looked at the cellular response to product toxicity by analysing transcriptomic, proteomic, metabolomic and lipidomic data. Focusing on the transcriptomics, you can select which comparison you want to make from a drop-down menu and choose the time point you want to look at, and Morph will draw a volcano plot of the data. You can adjust the fold change and p-values thresholds represented by the dotted lines. Morph lists genes above these thresholds, and clicking on either the gene name or a dot in the volcano plot opens the gene page where more details can be accessed. The gene page includes the location of the gene in the genome as well as the transcriptomic and proteomic data side by side. Transcriptomic data is also displayed in heat maps where expression patterns can be identified. Principal component analysis is used as a quality check to see if the data is grouping as expected and also identifies which genes contribute most to intergroup variance. The proteomics are laid out in the same format with the volcano, heat map and PCA. Morph also includes a flux balance analysis tool where a metabolic model can be uploaded, edited and solved to test engineering interventions in silico. The Genome Browser contains over 100 microbial genomes that can be viewed in circular and linear plots. These tools are free to use through the website. To find out how you can get more from your data, please get in touch.